Okay, I want to welcome everyone. This is the second FYI series for the IDGI group. So we are very lucky to have Brian Smith with us. He's an assistant professor here at DCRI. I actually have to bring notes because he has so many titles that too many to remember. He's also assistant professor in the Division of Neonatology and Quantitative Services over at Duke. He's a scientific advisor for the FDA, helps with designing clinical trials in pediatrics, neonatology, and also looking at data and doing data crunching. He's done extensive research with antifungals, antimicrobials in preterm infants, the sickest of the sickest kids. And we're lucky to have him here at DCRI. He's currently
joining
leading
conference
several of our clinical trials. So please welcome Brian Smith.
Thanks, Catherine. Well, thanks, Catherine, and thanks for y'all's attendance here this afternoon. And I'm just going to, what I'd like to talk to you about is sort of the lack of information on dosing for therapeutic agents in neonatology. And these are babies that are too sick to go home with the parents. They're babies that stay in the hospital and in the intensive care nursery. A large proportion of these babies are premature, and a large number are term babies that are very sick. And most of my research since I've been on faculty has been in the area of looking at dosing for these therapeutic agents. I've received research support from Astellas, Cadence, and Cubis Pharmaceuticals, as well as Pfizer. So I just wanted to start this talk by going over a case that happened when I was a fellow. So this, I think, was in around 2004, my first year of fellowship, so about 5 years ago. We had a baby that was a 25-week, 800-gram, actually a quadruplet infant, who had a complicated medical course, had several of the complications of prematurity happen to this baby in the first couple of months of his stay in intensive care nursery. And on about day of life 80, the infant looked sick, had blood and urine cultures that were drawn, and those cultures grew Candida albicans, which is a fungus that causes infections. In term babies, it typically just causes thrush or diaper rashes, but in premature babies, it causes bloodstream, urine, and infections as well as meningitis. And so the infant was started on antifungal therapy. After a couple of days, we started the baby on amphotericin B deoxycholate, which is the oldest antifungal that we use. It's been around since the 1950s. But the baby didn't do well for the first 3 or 4 days, and so we switched the baby over to amphotericin B lipid complex, which is a newer formulation of amphotericin B. That usually the advantage to it is you can give it typically in a higher dose, but that medicine didn't work either. The baby had cultures that remained persistently positive for Candida, so we added Fluconazole to the antifungal regimen, and that didn't work. Eventually, the infant received a different lipid product, liposomal amphotericin B. Also, it's about a month into the therapy, got caspofungin, which is one of the newest antifungals. And finally, after over a month of being persistently fungemic, the baby did clear the infection. The baby had a side effect, we think, from caspofungin, was switched over to voriconazole, another new antifungal, and then finally finished out a really prolonged course with amphotericin and caspofungin. We knew a little bit about the dosing of amphotericin B deoxycholate. We know a little bit about the dosing of ABLC and liposomal amphotericin B back in 2004. We did have some information on fluconazole, but it was essentially the wrong information, as we'll see a little bit later. And we had no information on voriconazole and caspofungin. And so here we had this very sick infant, and we were treating him with a whole bunch of medicines to no effect without knowing the right dose to half the medicines we were using. And in addition to not knowing the right dose for these medicines, we had no indication that any of these medicines work for Candida infections, and none of them are indicated for Candida infections in this population. Switching over to another therapeutic area, this is a graph of the number of infants that go home receiving reflux medications, so medicines like Zantac or Prilosec or Reglan, and these babies were born less than 1,000 grams. And as you can see, the percentage of these babies that go home on these medicines vary dramatically by what hospital those babies are born at. And so all these babies were really small at birth. You would, overall the mean birth weights were not that different between these centers. But you can see that in some of these centers, only 1 or 2% of the babies went home on reflux medicines, and this ranged up to 40%, and a couple of the centers, over 80% of those babies went home on reflux medicines. And these are also medicines for which we don't know the right dose of the medicine in preterm babies, and we do have some trials that have looked to see, do these medicines work, and none of them work. And so we have a medicine for which we don't know the right dose, and we don't, for all indications that we have, they don't work in this population, and yet a huge number of these babies went home on those medicines. Back to antibiotics. So this graph shows the proportion of infants that were born at 250 NICUs, and each one of these dots, the clear dot is the percentage of babies born at a NICU that received ampicillin and cefotaxime, and the black dot is the percentage of infants in that NICU that were receiving ampicillin gentamicin. So the very first pair of dots are on the far left-hand side, represent one NICU. And so in that NICU, 100% of the babies received Ampingent, and none of the babies received ampicillin and cefotaxime in the first 3 days of life. And essentially all the babies that are admitted to a NICU receive antibiotics on admission because they're in the NICU for a reason. They usually look sick, and we worry they're infected. As you can see, as you move across the x-axis, the number of babies that received Ampingent start dropping a little bit, and the proportion of babies that received ampicillin and cefotaxime increase. But the take-home message from this is that the babies are receiving the antibiotics not based on how sick they are, but really based on which unit they're in. So the babies that are in these 50% of the units, all of them are getting Ampingent. And the babies that are in this sort of 10% of the units, they're all getting ampicillin and cefotaxime. And so it's a unit practice. It's not looking at the baby to see how sick the baby is and whether or not they should be on one antibiotic or the other. And in practice, cefotaxime is a more broad spectrum antibiotic than gentamicin. And so typically or historically it's been used for sicker babies, and that ampicillin and gentamicin would be used for babies that aren't that sick. And again, you can see here the majority of these units really didn't use it like that. They just decided that we'll use Ampingent all the time, or amp and cefotaxime. And this is important because Danny Benjamin has a study from several years ago that showed that broad-spectrum antibiotics like cefotaxime are associated with the development of subsequent Candida infections later on, and that giving a baby a broad-spectrum antibiotic kills all the bacteria but allows for overgrowth of Candida and then predisposes that baby to Candida infection. So over the last several years, most NICUs have moved away from using cefotaxime in the first couple of days of life. But just a few years ago, this practice was common, and it wasn't really being used based on the baby. It was based on the unit. I think this is my last drug exposure slide. So this slide is to show you that the exposure to drugs really happens in the premature babies. These two medicines, dexamethasone and hydrocortisone, I chose to present here because dexamethasone use is associated with the development of cerebral palsy. And so we really don't like using this agent in premature babies. We use it to improve their lung function and to get them off the ventilator. And this was a widespread practice about 10 or 15 years ago. The use of dexamethasone has really decreased, but you can see that even as recently as a few years ago, a large percentage of the most extremely premature babies received this agent. I mean, up to a quarter of babies 23, 24, and 25 weeks gestation were exposed to dexamethasone, and we know its relationship to cerebral palsy. Hydrocortisone is another steroid for which we don't have very good follow-up data, so we don't know that it's associated with bad neurodevelopmental outcomes, but nobody's really looked in a systematic way. And you can see an even higher percentage of these babies have been exposed to hydrocortisone. As you get to babies that are less premature, so babies that are born 30, 32 weeks, a couple of months premature, very few of those babies are exposed to steroids, but babies at the limits of viability, a large percentage of those babies get exposed to steroid drugs. And then historically, just these are some examples of where we didn't know the drug was safe and we were using it in babies and disasters happen. So sulfasalazole is an antibiotic from the 1950s that people wanted to use because they didn't have to dose it every 4 hours and it was much more convenient to give. And then somebody decided to study it against another antibiotic in neonates to see which one did better, and they found out the older antibiotic was better and that more babies died with this newer antibiotic. And the reason they were dying is that the antibiotic displaced bilirubin from albumin and gave the babies kernicterus, and if the babies didn't die, they were severely brain damaged. Chloramphenicol was also another antibiotic from the 1950s. People like to use chloramphenicol because it had a really broad spectrum as opposed to penicillin. And then small reports started coming out about babies dying from just cardiovascular collapse. And then when this was looked at in a systematic way, the mortality of babies that got chloramphenicol was about 6 times higher than other antibiotics. And so it took about 10 years before people recognized a syndrome that we now call gray baby syndrome, from where babies don't metabolize chloramphenicol appropriately or like older children and adults, and at higher doses they die. And then more recently, as recently as the 1990s and the 2000s, Decadron, so the medicine we looked at on the previous slide, we were using it commonly to prevent chronic lung disease, to get babies off the ventilator so that they could go home. We found out that if this medicine was given to babies that weren't at that higher risk of developing chronic lung disease, those babies were at much higher risk of getting cerebral palsy. And in some populations, the number needed to harm, or the number of babies that if you gave a dose of Decadron that would subsequently develop cerebral palsy above the baseline rate was as low as 4. So if you treated 4 babies with Decadron, you ended up causing 1 extra case of cerebral palsy, and that's a really low number. So 30 years ago, the American Academy of Pediatrics said that it was unethical to continue to force physicians to treat each child that they treated with a drug as a trial with an N of 1, and that it wasn't only ethical, but it was imperative that new drugs be studied in children. And then 20 years later, 3/4 of drugs had insufficient labeling for pediatric dosing, safety, and efficacy. And this is in children, not neonates. As you go to neonates, the number is even worse. And if you go to preterm neonates, that percentage is probably above 95%. So in order to make the situation better, there were a series of legislative acts designed to increase the study of drugs in children. In 1997, the Food and Drug Administration Modernization Act established pediatric exclusivity, which allowed for sponsors to do studies in children in response to an FDA written request. So the FDA would write a document that would say, if you do these several studies in children, and you submit them to FDA and submit the data to FDA, that sponsor would receive a 6-month extension on their patent. And this generated a lot of studies in children. 5 years later, the pediatric exclusivity mechanism was renewed. And then the Congress also established a mechanism by which a collaboration through the FDA and NIH could stimulate pediatric studies on off-patent medicines. And so, of course, pediatric exclusivity didn't stimulate any trials in off-patent medicines because you couldn't give the company a 6-month patent extension. And then in 2003, in the Pediatric Research Equity Act, the pediatric rule was written into law, and that allowed FDA the power to make sponsors do studies in children in new drugs, even if they weren't going for pediatric labeling of that drug. If it was a drug that was going to have substantial use in the pediatric population, the FDA could require sponsors to do trials in children before the drug received marketing authorization. Unfortunately, this rule doesn't have enough teeth in it to make the sponsors finish the trial before marketing authorization occurs. And then finally, in 2007, both PREA and BPCA were renewed for another 5 years, and so they'll both be in place until 2012. The European Union was a little bit behind. They really didn't get started with their legislation until 2006, But in 2006, they developed a mechanism by which sponsors had to come up with a pediatric implementation plan before a drug received marketing authorization. And this would be for any new products, new indications, new formulations, or new routes of administration. The PIP outlined the studies in pediatrics that the sponsor had to complete before the drug received marketing authorization. And so if this was a drug for which the sponsor was only going for labeling in adults, if the sponsor did not hold up their end of the bargain and complete the studies that were outlined in the PIP, they didn't receive marketing authorization. And so this legislation had the power to force the sponsors to get the trials done. And then if once all the trials were submitted to the EMEA, the sponsors there also receive a 6-month patent extension once all the trials are completed. This is just a quick graph to show you that in the European Union, the PIPs typically get approved at the end of phase 1 testing in adult patients. And that in the FDA, under the FDA system, the trials that are developed under PREA typically occur later, towards the beginning of phase 3 testing in adults. In the European Union, the pediatric trials undergo a pediatric implementation plan compliance check. If those trials aren't done at the time that the sponsor is going for marketing authorization, they don't receive marketing authorization. Under PREA, it doesn't work quite that way. They can still get marketing authorization even if they haven't completed the pediatric trials. So because the pediatric legislation in the European Union and the U.S. overlap a lot, the FDA and the EMEA have begun a collaboration several years ago where they talk by monthly teleconference. They share PIPs over to the European Union sends PIPs over to the FDA. The FDA shares written requests with the EMEA in order not to overlap requested studies from sponsors. The DCRI actually got involved with a collaboration with the FDA back in 2005. Danny Benjamin and Jennifer Lee were asked to be scientific advisors within the Office of Pediatric Therapeutics. And currently, myself and Mickey Cohen-Wolkowitz have that same position, where we analyze data from pediatric studies generated from this legislation on the previous slides. And some of the projects that we've worked on while we've been there, we've looked at the publication rates of trials that were submitted for pediatric exclusivity. So even when sponsors went to all this effort to do these trials in pediatric patients, a large number of them were never published in the peer-reviewed literature. So close to 50% of those trials aren't available to pediatricians that are taking care of the patients. Jennifer Lee and Danny looked at the economic return for sponsors that were completing trials for exclusivity. So they calculated the cost of the trials outlined in the written request, and then calculated what economic impact on the sponsor that the 6 months patent extension would have, and found that of course most of the trials, most of the sponsors, the economic impact was fairly substantial. We've also looked at a couple of safety issues that came up. FDA, for drugs that receive exclusivity, the 6-month patent extension, the FDA has to do a 1-year safety review of all adverse events that are reported to the FDA. And under that piece of legislation, they have actually made several changes to the label and have also issued med guides for several of these drugs when they've gone back and reviewed the safety data. We looked at the safety of giving children placebo in the context of a hypertension trial. There are some ethical concerns that it's not ethical to give a child a placebo when they have a disease, so hypertension. And then we found in short-term antihypertensive trials that adverse events were no higher in the placebo group relative to the treated group. And then finally, there were several antihypertensive trials that were submitted to FDA that failed. And these are drugs that work in almost every population that they've been given to in adult patients. And they were given to antihypertensive teenagers, and they didn't work. And Danny looked at some of the reasons these trials didn't work and found that endpoints were important and not necessarily the size of the trials. In fact, of the 6 trials that were submitted, the 3 largest were the failed trials. The 3 smallest actually reached their endpoints. So after all this legislation and all this work and all these studies, neonates still remain understudied. There's been 376 written requests, 167 drugs have received the 6-month patent extension. There's been almost 160 pediatric-specific labeling changes, and if you include the trials under PREA, that number is over 300. The off-patent mechanism from FDA and NIH is a little bit behind. No labeling changes yet, but we hope to change that with a trial that was led here at DCRI. But neonates have been left out. So if you look at the first 7 years of BPCA, there were 121 drugs, 280 studies in children, and over 80,000 children were studied. If you limit this to just the studies that were performed in infants, these numbers go down by an order of magnitude. So there were only 17 studies, 17 drugs, 22 studies, and under 3,000 infants. If you exclude topical agents, creams, eye drops, antihistamines, which we never use in the NICU, Claritin, Zyrtec, and antiretrovirals, which besides AZT we never prescribe in the NICU, you're left with 5 drugs, 6 studies, and less than 200 infants studied. And these are those 5 drugs. So the first one is ranitidine. It's an H2 blocker. The rank column comes from data from 250 NICUs over a 10-year period. So it was the 18th most prescribed drug in the NICU over that 10-year period. There was one study that enrolled just infants, and none of those infants were premature. And as we saw from one of those first slides, in some centers up to 80% of infants go home on reflux medicines, and Zantac would be probably the most common of those medicines. Famotidine is another H2 blocker. We almost never use it. It was the 153rd most commonly prescribed medicine. They did enroll almost 60 infants, but none of them were premature. Linazolid's an antibiotic. We do use it in the NICU, but not that often. But they did happen to enroll 18 premature infants. Remifentanil was the 4th drug. In that 10-year period, it was used never in those 250 NICUs, and the trial enrolled no premature infants. And then finally, nizatidine is a 3rd H2 blocker, which is uncommonly used in the NICU, and in these 250 NICUs was never used, and they enrolled no premature infants. Moving to antibiotics. Antibiotics are the most commonly prescribed medicines in the NICU. So the 1st and 2nd and 3rd most commonly prescribed medicines are ampicillin, gentamicin, and cefotaxime. So again, there's a column that shows the rank in which that agent was used in those 250 NICUs. In the last column are the number of infants for which PK data is available in the literature. And even for ampicillin, which is the most commonly used agent of all agents, not just antibiotics, in the NICU, there's only a handful of infants that are very premature for which data is available. There's no data available for nafcillin, metronidazole, cefazolin, Meropenem. There's one infant that was 27 weeks, and for piperacillin tazobactam, there's none. And this is just an example of some antimicrobials for which no data is available. Moving to antifungals, so in 2004, this was the number of infants that were available in the peer-reviewed literature for which PK data was available. There was a little bit on amphotericin B-deoxycholate, which again is a medicine that's been around for, I guess, almost 60 years now. The lipid product, amphotericin B-deoxycholate, ABLC, ABCD, there's a few infants. Liposomal, there's a few infants. The azoles were medicines that came out in the 1980s and 1990s. There's a little bit of data on fluconazole, and we'll look in a slide in just a moment where they actually recommended the wrong dose. There's no information on voriconazole, and in 2004, there was no information on the echinocandins, which are the newest class of antifungals. So why is it important to study the dosing in neonates? So the dosing for many agents is likely to be very different than it is in older children and adults. And these are just a handful of reasons why this may be different. So infants and neonates don't secrete much acid in their gut. And so if you're giving them oral medications, that often affects the absorption. Absorption is also affected by gastric motility, which is decreased in newborn babies, but it's almost non-existent in very sick premature babies. Biliary circulation, which is important in drug metabolism, is very different in neonates. And then for topical agents, they have a really large body surface area, and in preterm neonates, their skin is very thin, and so absorption is really increased in newborn babies. Drug distribution is very different in neonates. Their water content of their whole body is drastically increased compared to older infants and children. They typically bind drugs less well with their plasma proteins, and this means that often the therapeutic exposure to drugs can be higher because much of the drug is free. And then there are big differences in where blood flow happens in babies. And so the blood flow to the kidneys, the blood flow to the liver, the blood flow to the intestines is very different than it is in adults. The enzymes that break down medications in the liver are typically not as developed in babies as they are in older children and adults, although some enzyme systems are actually reversed. They're increased in babies. And then finally, the GFR, the urinary output of preterm babies, is really decreased compared to older children. So some of the reasons that we ended up with no data on babies for most of the therapeutic agents include the fact that the studies are hard to do. And they're hard to do because infants don't have much blood. And so PK trials typically require a fair amount of blood. Not that long ago, many assays required a large volume of blood, 3 cc's or 5 cc's or more. Preterm infants are typically really sick, and so people had a hard time designing trials and carrying out trials in very sick preterm babies. We didn't have assays that could use low volumes of blood, and there aren't a lot of pediatric pharmacologists to analyze the data. There are ethical concerns with enrolling babies and children that can't consent for studies, and psychologically it's hard to come to a parent with a consent form and say, can I do an experiment on your baby with this drug for which we don't know the right dose or whether it's safe. And so consenting for these studies is extremely hard. And then just as an example for the blood volume, here a 500-gram 23-week infant is about the smallest baby that we take care of. We do take care of some babies that are 400 grams or above. But the total blood volume for that size infant is 42 milliliters, which is less than 3 tablespoons. And then compare that to an adult who has 5 liters of blood. And so if you take 10 3-milliliter PK samples, and 3 milliliters is less than a teaspoon, you've used up 3/4 of their blood volume for those 10 samples. And so that's not even counting, most PK trials you're getting lab values and safety laboratory values. So you quickly run out of blood to do the study with. So moving back to fluconazole, this is just an example of where we did have a little bit of information. Unfortunately, it was somewhat misguided. From a publication from 1998, the authors recommended a dose of 4.5 to 6 milligrams per kilo per day. Another author a few years before that had recommended 6 milligrams per kilogram, but to spread the dosing out in the first couple of weeks, every 72 hours or every 48 hours. And then if you read the FDA label, if you still read the FDA label, the recommendation is 12 milligrams per kilo, spaced out every 3 days in the first 2 weeks of life. So Duke was involved with a study that was recently published where 55 infants were enrolled, and these infants were already getting fluconazole as standard of care. And they had samples either drawn or scavenged samples obtained from the lab that were left over. And almost 360 samples were obtained from these 55 infants. The infants were receiving anywhere from 3 to 12 milligrams per kilo per day, and all the 12 milligram dosing was actually here at Duke. A large percentage of these babies were the smallest infants in the unit. We don't know exactly what exposure to fluconazole infants need, but in adults, the goal is an AUC, which is a measure of exposure to drug, of 800 in the sickest adults. And the results from this trial showed that among the premature babies, so the solid line is the 23 to 29 week infants, getting 12 milligrams per kilo per day did not ever reach the goal of 800, which is the goal in the sickest adult patient. So even after a week of therapy, their line never got to 800, and it took several days for it to get above 400. And 400 is the goal for adults that aren't that sick. And again, we don't know what that number should be in babies, but you would think it would be somewhere between 400 and 800. And it's a little scary to think that even with sort of the highest dose of fluconazole that was being used in this study, it took several days for those infants to reach that exposure. And so what this data would seem to suggest is that babies might benefit from a loading dose the very first day of therapy, so to double the dose. And that's actually the common place in adults, is they get a double dose, a loading dose on the first day. For mycofungin, again in 2004, we knew nothing about mycofungin. There was a study published in 2006 that gave babies anywhere from 0.75 milligrams per kilo to 3 milligrams per kilo, and found out that the serum levels were really low with mycofungin. Adults receive approximately about a 2 milligram per kilo dose of mycofungin. We know in babies that when babies have Candida infections, the Candida is commonly found in their brain. And so they have meningitis or they have abscesses within their brain. And so it's important that if you're treating a baby with antifungals, that the agent be able to get in the CSF. And so using a bunny rabbit model for Candida meningitis, William Hope and Danny found that it required doses of up to 15 milligrams per kilo to kill the Candida. And this shows you that in the bunny model, the Candida is found in all these compartments within the brain. And then this slide just shows you that as you increase the dose of mycofungin from 3 milligrams per kilogram up to 15 milligrams per kilogram, you kill Candida. And so as those bars shift over to the left, means you've increased the killing of Candida. And you can see that those bars keep shifting even as the dose approaches 15 milligrams per kilogram. So based on that previous trial that used the really low doses of mycofungin and this data from the rabbit model of Candida meningitis, we led 2 trials of mycofungin pharmacokinetics here at Duke, where we administered doses between 7 milligrams and 15 milligrams per kilogram to premature sick infants. And we found that the exposure at these levels was about double what's seen in adults, and this may be the appropriate exposure. We still don't know the answer to that because, again, babies typically have or are commonly affected with Candida infections in the brain where you don't have that problem in adult patients. So 5 years later, we have more information in fluconazole than we did before, and we know that 6 milligrams every 3 days is not the right dose, and that 12 milligrams every day is probably the bare minimum right dose, and that babies may need a loading dose of fluconazole. And we now have, there's been a publication that used a dose of caspofungin, and there's been the trials here at Duke with mycofungin. So the antifungal studies that we have going on right now, we have a loading dose fluconazole study. So we're evaluating the safety and PK of giving babies a high dose on the first day of fluconazole. We're also looking at Anidulafungin, which is the third echinocandin, and then DCRI is the coordinating center for a large study of fluconazole prophylaxis. And so there's been, we'll look on the next slide, there's been several studies that suggested that giving premature babies fluconazole as a prophylaxis for Candida decreases their risk of getting Candida infections. And so this is a 360 infant study led by Danny Benjamin, and it's ongoing now in 30 centers. And so there's been 2 previous placebo-controlled randomized trials that showed fluconazole works, but most NICUs aren't using it. And the reason they're not using it is those previous trials, the rate of Candida in the placebo group was extraordinarily high. And in most US NICUs, the rates of Candida aren't that high. And so most people didn't feel like those trials would apply to their NICU. And there was also a lack of neurodevelopmental follow-up. And so it's important when you're giving babies a therapeutic agent that you know what it does to their brain long term. So it's not just important to prevent a disease or to make them survive, but it's also important to protect their brain. And so we have no information on whether fluconazole prophylaxis affects the baby's brain. And so that's the reason we have this trial that's ongoing. It's a randomized placebo-controlled blinded study, 360 infants. The babies are going to get fluconazole at a prophylaxis dose, 6 per kilo twice weekly, up to study day 42. And the primary outcome for the study is death or Candida. And we are going to follow the infants for a year and a half to assess their neurodevelopment in the survivors. And the study is about a third of the way through enrollment. For antibiotic studies, actually this one's not ongoing. DCRI was involved with, and Danny Benjamin led, the Meropenem study for infants. And this study was funded through the NIH FDA mechanism under BPCA. And so this was a 20-center study. 200 infants we completed enrollment 2 weeks ago. It was a PK safety with a little bit of an efficacy component. It wasn't randomized. It was an open label study. And we also had several sub-studies. We are going to look at the CSF PK of meropenem. We're going to look at the utility of scavenge sampling, so getting samples out of the lab that they're going to throw away. We're going to look at the utility of that. And then Mickey Cohen-Wolkowitz is going to look at the distribution of meropenem in the intestines. And this study was actually looking at meropenem in infants that were having intra-abdominal infections. Again, this was a 20-center, 200 infant study. And the inclusion criteria were infants less than 91 days of life that had an intra-abdominal process going on. So either they had a disease called necrotizing enterocolitis, which is sort of a catastrophic infection of the intestines where the intestine dies. Those infants were the population we were studying. We planned in the study to start the infants at these doses that are in bold. It was based on both their gestation at birth and how old they were since birth. Infants that were older, we predicted would metabolize the drug faster, so they got a higher dose. We did an interim PK analysis to see if we needed to give the babies actually a higher dose of meropenem. And what we were looking for in the interim PK analysis was the concentration of the drug. So this would be when the dose is given to the infant, and the dose in the plasma peaks, and then over 6 or 8 hours, the plasma level comes back down. And so we were concerned with what is the concentration of the drug 3/4 of the way through that dosing interval. So if the dose was given every 8 hours, we wanted to know what was the concentration of meropenem 6 hours through that interval. And the reason that's important is for drugs like meropenem, the time the drug spends above a certain level determines how well it kills bacteria. And so, again, this is what we were trying to calculate in the PK analysis. And what we found was these are the 4 groups that we enrolled in the study, and the goal was to have everyone above this level of 2 micrograms per milliliter, and we only had 1 infant in 3 groups below that level. And based on our rules for advancing the dose, we did not advance the meropenem dose in this trial. And so the levels that we chose for dosing meropenem at the outside of the trial ended up being the doses I think that we're going to recommend based on the infant's gestational age and their postnatal age. These graphs just show how the clearance of meropenem from the body is related to gestational age and postnatal age, so how many days the baby is old. But it's even more closely related to the corrected age, which is a combination of the infant's gestational age and the postnatal age. So an infant born at 24 weeks who's 2 weeks old is really 26 weeks. And so when you combine those two, it seems that the clearance of meropenem was really sort of tightly related to that factor. So from the studies that we've completed so far, what we've learned about dosing for a few agents is the right dose for fluconazole is not 6 milligrams per kilo every 3 days. It's 12 per kilo per day, and they might need a loading dose. For meropenem, we know that the dosing is related to gestational age and postnatal age. For mycofungin, the right dose is likely not 1.5 milligrams per kilo or 2 milligrams per kilo. It's probably at least 10. For Anidulafungin is a study we actually have ongoing right now, but we have some preliminary data that shows that the adult exposures, the exposure to Anidulafungin is very similar to what you see in adults. The antimicrobial studies that we have funded or planned, we have a single center study in cefazolin, a single center study with daptomycin. DCRI is going to lead a piperacillin tazobactam study that was funded through the CTSA. That's going to be a 4-center PK study. We're going to further look at the metabolism of mycofungin. We plan to look at a higher dose Anidulafungin regimen, and then Mickey Cohen-Wolkowitz, who is in our group, will also be looking at metronidazole, another antibiotic. So I think what my take-home message would be for neonatologists and pediatricians is that if you think you know the right dose for the patient in front of you, you're probably wrong. You're probably just using the dose you used last week. And that we know the least about dosing safety and efficacy in premature infants, and that we know almost nothing, especially when you look at efficacy. And then when we do bother to get around to studying drugs in children, we end up being surprised with the drug's dose, the fact that the drug may or may not work, and then finally that even if companies or people bother to study drugs in children, the results of these studies are often not available to clinicians. You can't just go to PubMed and pull up that study if it's not published. And I would like to thank Danny Benjamin, who's been my mentor through fellowship and now as I'm on faculty, Mickey Korn-Wolkowitz, who's on faculty now in Peds ID, and then in my division of neonatology, Ron Goldberg and Mike Cotton. And I'll stop there and take any questions. Sure.
I have two questions. The first was related to the PK. How many PK samples are you typically taking in these studies, since you said just 10 wipe out 70% of their blood?
So fortunately for essentially all the drugs that we've studied so far, an assay has been developed that uses 100 microliters of plasma. So that's 0.2 mLs instead of 3 mLs. And we usually write in the protocol up to 6 or 7 samples per baby. At Duke, we typically try to get those samples when the baby's already having labs drawn for another reason. Unfortunately, in neonatology, babies don't have central lines that draw like they do in adults and children. They get heel sticks for their labs, and so if a baby's getting 6 samples over 2 or 3 days, they may have 4 or 5 lab draws during that period. And so we have enough flexibility in the protocols to, allow the nurse just to, squeeze the heel one more time to get that sample. So usually the babies in our studies maybe get poked for the study once or twice at the most.
Okay. My other question, based on your slide before this, so what are doctors following?
So they're, despite the fact that nobody knows what the right dose is, there are a lot of publications out there that use dosing developed in older children and adults. Mm-hmm. And so, I think most NICUs use Neofax, which Barry Mangum is one of the authors of that publication, and they do the best they can. If if there's no drug study in babies, they use dosing that was done from studies in usually older children or older infants.
So you did a compelling job on why we need to do more studies, and so I have 2 questions. One's for you for sure, and that is under the BPCA trial that we just completed enrollment. The team at DCRI was able to better the timelines of any other group that's in the mechanism. We're actually the first trial to be completed under that mechanism, almost on time and on budget, which is phenomenal for a government-sponsored trial. And as many of the staff know, the PI for that trial, which was me, was often maybe less than available, which is not uncommon here at DCRI for some of the more senior faculty members. So can you provide the staff, and I'm also interested in hearing from the staff members here who were on that trial. I see that several are in the audience. What were some of kind of the operational challenges from a multi-center perspective? We worked with an outside coordinating research organization. We worked with NIH. What were some of the challenges in working with some industry people, some government people that we had to overcome? And then I'm also interested in hearing from the staff members who worked on that trial, kind of what they viewed as some of the bigger, from a multi-center standpoint, some of the bigger operational hurdles that are unique to the baby studies as opposed to the adult studies.
Well, the one offhand I can think of is for the studies for BPCA. They contract with an outside CRO for all their studies, and so there's 3 or 4 studies under BPCA, the NIH FDA collaboration. They use the same CRO, and it's not DCRI. And so I think from our standpoint, the leadership team here at DCRI, I think it may have been easier had it all been here. And so we were already doing a lot of things related to the trial, and then we had a separate CRO, and then we had the NIH, and then we had the sites, and so it was sort of 4 different parties as opposed to 3 if all that would have already been here at DCRI. Since the PI was already here at DCRI, that would have been very convenient, I think. Going into the trial, we know that enrollment in babies is really hard, and so we made the inclusion criteria for the study extremely broad going into the study, and I think that was probably some of the problems with maybe some of the other studies under the same mechanism, is that they were complicated studies, the inclusion criteria were somewhat complicated, and that enrolling children, and especially neonates, is hard, and so to keep the inclusion criteria as broad as possible, I think helps enrollment. So I don't know, those 2 things off the top of my head. I don't know, Catherine or Jamie wants to.
Jamie has. It's all Jamie.
I have 2 questions. When you do a study, you do look safety issues, and some of the events are based on people's feeling, like headache or something like that. And since this population, they cannot speak for themselves, and would that affect the safety profile for a study?
It does. And to me, I'm still learning this aspect of the trials. In neonates, getting safety data is extremely hard. I think it's hard in adults. It's pretty hard in children. And it's extremely hard in neonates, and it's really hard in preterm neonates that are really sick, because bad things happen to them on a daily basis, whether you're giving them an experimental drug or not. And so it's really hard to get a signal of, is something bad going on in this population, because of the babies we enrolled, the 200 infants, lots of them had seizures. A number of the babies died. A number of the babies, their intestines perfed, or they had head bleeds, or whatever. So they have these really catastrophic things happen to them during the course of the trial. But you would need just a tremendously large number of infants to enroll and compare against another antibiotic to see if there's a difference in those outcomes. So I think safety outcomes are hard to look at. In this trial, we were specifically interested in seizures with Meropenem because a drug in the same class, Meropenem, is associated with an increased risk of seizures. And so we had some sort of special case report forms and safety committees that were built to really look at that outcome specifically, because that's what the NIH and FDA were interested in. But even that, I'm in a non-randomized trial, an open-label trial, I'm not sure you would ever be able to say that, yeah, this drug's associated with seizures or not. I mean, we'll be able to say the percentage of babies that were on the drug in the trial that had seizures, but it would be hard to compare that number to a similar group of infants.
Yeah, my second question is about the consent. And if the mom is underage, and for the consent, does the consent form need to be signed by the mom and somebody else need to sign, or the mom?
So that's a good question. So even in the state of North Carolina, it's different. UNC does the exact opposite compared to Duke. So at Duke, if the mom is unmarried and under the age of 18, the maternal grandmother signs. That person has custody of the baby. And at Duke, we take that to mean that that's the person that needs to sign the consent for that baby, not the 17, 16, 15-year-old mom. At UNC, they're the opposite. They do the opposite. If the mom's 12 or 13, they typically would get the maternal grandmother. But a mom that's 15, 16, or 17, that's who they get to sign the consent. So my guess is in these 20 sites, there were probably 6 or 7 sites that got the consent from the 16 or 17-year-old mom, and there were a dozen sites that did like Duke and got the consent from the maternal grandmother. Sure.
I see that there's some financial incentive for pharmaceutical companies to get pediatric data. Is there any kind of financial incentive for diagnostic PK companies to refine their assays so that they will require less blood, or are you working with LabCorp, the Canapolis group, to develop anything like that?
So that's a great question, and it's a little bit outside my area of expertise. It seems like all the drugs that I've been involved with, there has already been an assay, either because the sponsor knew they were going to study the drug in children, and they worked to develop that. Danny may be able to answer that better than me.
Moderate. Would you like your participants to be able to unmute their own lines by pressing *6? Press 1 for yes and 2 for no.
So
all participants have been muted. You will now rejoin the conference.
So so two things. So Brian's right. A lot of times now companies, when they work with us, they're willing to go and pay for the assay to be at done smaller, but they do that as part of the agreement with FDA, in that FDA says you will get the study done in babies. And so, which is rare, but the few times that they do, the company will do it. The second time that they'll do it is if anonymous thought leaders call up the company and say, we'll do this for you, but we're only going to do it if you do the micro assay for us, okay? And puts external pressure on the company. And the third way is for us to do it ourselves. So Brian and Mickey are doing a lot of that work in collaboration with like Case Western Reserve or UNC, where they take money off of their own grant money, or they take money off of consulting, and rather than taking that money personally, those guys set the money aside for labs to develop micro assays. So they do it kind of nights and weekends style. And then, you know, as far as public pressure is concerned, you know, we had a trial with a company where they refused to do the study in babies because they would be, you know, developing that assay would be too expensive. And so we called up the head of, is this being recorded? So when we called up, I won't tell you the name of the company, I won't tell you the name of the VP, but we called up the VP of Global Operations and said, we're going to name this study the 137.1 study. And he said, why is that? I said, well, we're going to name it that, and we're going to put that in the title because it costs $137.10 to buy your product off the shelf here at Duke University, which is available to these babies. And since you aren't supporting this study, when we publish this is going to be the Duke 137.1 study. And he was somehow able to manage shaking down some money from within the company to get the assay. So it can be done. You just have to know where and how to apply pressure to the companies, and have to be willing basically to really go to the mattresses for it.